Our next Gogec speaker is Dr. Seok Moon, Associate Professor at Washington University, St. Louis. He did his undergrad in chemical technology at Seoul National University, got some industry experience at LG Life Sciences before doing his graduate studies with Christella Prather at MIT, postdoc with Chris Voigt at UCSF and MIT, before starting at WashU. His work spans foundational SynBio tool development like RNA regulators and general engineering tools for Rhodococcus, which support metabolic and circuit engineering for what I think is fascinating applications in plastic upcycling and smart probiotics. Let's hear what he has to share with us.
Okay, can you hear me okay?
Yep, we hear you.
Okay, I didn't have the ability to share, but I guess I have now. Do you see my slide okay, or you see my presenter mode?
We see your proper slides.
So it's going forward?
Mm-hmm.
Okay, great. So hello, everybody. My name is, again, Seok Moon from Washington University in St. Louis, not Washington, D.C., or not in Seattle, Washington State, or not an alien from the moon, although my last name implies. It's my great honor to present today because this is one of the best conferences to me. I mean, one organized by young people, our future leaders. That is probably the only one in the world. I love it. I originally want to talk about my two projects in detail, but decided to give a general overview instead, considering potentially broad audience. Let me introduce my university and St. Louis with some photos and videos first, because I mean, many people kind of thinking I'm living in Seattle or somewhere else. So as you know, St. Louis is very well known for the baseball team Cardinals. As you may know, it was the place for the Olympic game. It was the first time that the Olympic Games were held outside Europe. In 1904, the game was actually held in Washington University, my university. Also, it is known due to Budweiser, the world famous beer, and you can visit the brewery featuring the factory of beer museum and free beers. So why not? And not interested in sports and beers? If not, we have one of the largest urban parks in US, 2 times bigger than Central Park in New York City. All museums are free, and outdoor Broadway musicals are also free for many seats if you don't mind sitting too far. Also, Sigma Aldrich, the main headquarters here, Monsanto Bayer headquarters, of course, the notorious but global company also here in St. Louis. Also, my lab alum, Tatenda Chopra, PhD student in my lab, actually after graduation, joined Pfizer in St. Louis and developed and produced Pfizer COVID vaccine here, basically saving us, entire world. And my university is also affiliated with 25 Nobel Prize winners, and university ranking is 22nd in the world. More importantly, 3rd for best college food. And what else do you want to know? In fact, President Obama declared pie pizza at St. Louis is the best he ever had, and after winning the election, even invited the owners of the pie pizza to come cook their pizza for him in the White House. We also have $20 sushi buffet restaurant, and of course, Midwest barbecue. In 2014, there were some bad incidents called Ferguson riots, but that location is not close to the campus, and the areas near the campus is relatively safe. I stroll after dinner with my wife at night. I see rabbits all the time. I want to play this one if that is working. So there are two rabbits, probably couples. Then they kind of do something, and then one run away. Then the boy too sad. And also, I'll play this another guy. I also always see fireflies in the summer and night during my stroll. I'm not sure you could see this one. There are but lots of small dots, flashing temporarily, and that's kind of oscillation. I believe the more than 10 fireflies actually here in this picture or movie. So my point here is, my area is relatively safe and also clean, very clean. Those kind of fireflies do not, survive in the contaminated area. Okay, so I will start with my dream. My dream is engineering biology. Our ancestors dream of flying in the sky, but instead of creating birds or transforming themselves into birds, they analyze birds, design wings and propellers, and assemble them together to make airplanes. I want to take the exactly same approach. Instead of creating completely new cell, including yeast to make beer, I analyze existing cell, design synthetic DNAs, and assemble them together to make engineered cell that can be used for biochemical generation, biomaterial production, and even for some medical and environmental applications. Of course, we call it synthetic biology. I want to talk about the synthetic biology history first. We learn first to see the letters, and then to read the letters, words, and sentences. Now we are learning how to write poems. And we took one less traveled in this photo, as you can see. And for your information, this photo's public release was approved by Kristalla Preder and my PhD advisor. Actually, this is her favorite photo. Actually, I didn't make this one. This one made by some magazine. Okay, let's move on. When we come to seminars like this, we all use our sensor, eyes and ears, and then we perform computational operation by asking whether the slides are good and the speaker's voice is comfortable. And then we decide our action, something like this, and I cannot check whether you are doing this one, or attention to the top. Bacteria do the same things. Bacteria continuously monitor their environment, process signals, and decide their actuation. Here's my question. What if we can combine bacterial sensors, circuits, and actuators in a human-defined way? If that is possible, we can use bacteria for our goal from creation of tumor-killing microbes to toxic chemical degrading bacteria. My first topic is probiotic engineering. I envision our futures as follows. We engineer microbes that sense medically relevant signals and respond to them for diagnostic or treatment applications. I'll give you a bit of the history of my lab before moving forward. This sense and kill pathogen concept was my job talk idea in 2011, 10 years ago. Unfortunately, or fortunately, I saw a poster about this unpublished paper at SynBYSS 5 conference at Stanford in 2011 summer, right before the Voigt lab, my postdoc lab, moved from UCSF to MIT. If we moved to MIT before the conference, I might not have attended that conference because of the hectic, moving. And I am still wondering how my career would have changed, because I probably talking about this idea during my job interview. For your information, Ron Weiss at MIT, of course, a very famous synthetic biologist, was good by this paper because they are doing exactly the same in the project when this paper is published. Luckily for me, I finished all the experiment before the moving because moving is kind of disrupting. Due to the move, I have plenty of time to write my Nature paper starting August 2011 or July, I believe, while waiting for equipment, etc. Unfortunately, due to the moving, Chris Voigt, my advisor, did not have time to review my paper, so my CV for the job application kept changing. For example, my October application package says this paper will be submitted soon, and then my December CV says the same. So without this paper under review or published, I went to job interviews and luckily got my position at WashU. And for your information, I believe we did submit this paper to Nature in February, I believe. And then we published this paper 2012, the same year after I joined the WashU. I timely changed my job talk idea and I got my WashU position, as I said, 2012. And this was my first proposal I wrote when I started my position, because I kind of really wanted to solve the global problem, including some problem that actually prevalent in Africa. And actually at that time, Bill Gates actually highlighted this project with the 4 others on his Facebook among 60 grants selected in this round. And of course, what an honor. Unfortunately, I did not and do not still have Facebook account, so I didn't know this posting. And actually my friend let me know about it much later when this poster and this posting is kind of out at the time. I remember in also 2014 at the first SEED conference, that's one of the most famous synthetic biology conference. And actually I presented this project that is still 1 year kind of progress, so not that much data. So I kind of present lots of idea, including kill switch and biocontainment, probiotic kind of curing our body disease and so on. I present that one right after Frances Arnold talk, her keynote talk. And as you know, Frances is my academic grandma. and Nobel Prize winner. And of course, I was so nervous at the time. And I literally looked at her right before my presentation, and I was frozen, and I do not remember what happened afterward. Even though I started my probiotic project in early 2013 with the idea inception in 2012, I had not received the follow funding for engineering probiotics until 2017, when I got ONR Young Investigator Award, followed by NIH R01, and also USDA EPA very recently, while I got multiple grants from NSF and DOE for relevant preliminary but fundamental study for this probiotic work. So I was scooped by many big names, including, of course, Jim Collins, my hero, and also Pam Silver and some others. Well, nothing is too late. And then my paper actually, one of my papers actually published last year in Cell System. And also my paper is right now under review. We kind of submitted one paper, one of the top journal, and with the very minor revisions. So that paper will be out hopefully this month, including some kill switch ideas. I mean, we kind of originally thinking about 9 years ago. So please stay tuned. So I mean, because I mean the paper already out, another paper will be out soon. So I'm not gonna talk about in detail because I have limited time today. But my first subtopic is basically sensor development. One of the main challenges was lack of well-characterized application-relevant sensors. For example, we had used lab chemical inducible sensors for toy system demonstrations, but we cannot use these sensor or chemical in the human body because this might be toxic to human or not available in the human body or expensive. Considering the real world situation like the short pH pulse in the stomach and then aerobic outside to anaerobic inside the gut transition and temperature shift when the bacteria are released from the body, that is the 37 Celsius, of course, and outside temperature a little bit cooler. I mean, depending on the area you're living in, even Africa, temperature at night, below 37. Basically, we consider those kind of real world situation, and my lab actually developed many sensors for pH, oxygen, temperature, and so on since 2012. So these are the list of those sensor and paper we published, and I'm giving some example. Also, we developed biogeographical sensor that would activate only in the large intestine, not small intestine, thinking about the chemical composition difference. And additionally, my lab also developed various pollutant sensors for bioremediation application. However, developing medically relevant chemical sensors is still lagging. Other labs have recently developed or used sensors and circuits for various medical applications, as we recently reviewed, and some selected examples are shown here. Despite these recent advances, ligand-specific sensing is critical, and still very challenging, especially when sensing and responding to structurally similar chemicals with distinct biological outcomes. For example, phenylalanine and tyrosine are structurally very similar, only one difference, oxygen or no oxygen, but associated with the distinct disorders. For example, phenylketonuria, if you have too much accumulation of phenylalanine, and that's a very critical disease, especially baby, and sometimes lead to death, or tyrosinemia, that is about the tyrosine accumulation. Similarly, phenylethylamine and tyramine are neurochemicals, believe it or not, actually found in the gut or some food with the distinct biological outcomes. So differentiating and then detecting specifically one chemical versus the other is very, very important, because this might be lead to misdiagnosis, and that is the critical and prevalent kind of problem in hospital. And actually, I am one of the victims of misdiagnosis that kind of ruined my previous years, suffering from some side effect of the drug and so on. Let me introduce our neurochemical sensor. Notably, we employ a dual knob system consisting of amine neurochemical converting enzyme, in this case, TIN-A, that making the aldehyde. And also, we also use corresponding aldehyde binding transcription factor, in this case, FER, in this figure. We confirm this dual knob sensing mechanism by showing that the TIN-A enzyme is required for the function. Interestingly, TIN-A and FER homologs are found in many human intestinal bacteria, especially E. coli, but not in our host Nisl strain, the probiotic strain, enabling orthogonal sensing. So in summary, for the first part, without showing the detailed data due to time limit, also this paper is already published, I'm saying we developed for the first time specific sensors for structurally similar amino acid and also urochemicals, laying the groundwork for developing medically relevant probiotic sensors. The paper actually available since November 11. So just please check it out if you're interested in more detail. Additionally, we demonstrated a generalizable method of protein engineering that is time efficient and require small library size with only a basic understanding of protein structure. So at that time we started this project, unfortunately, we did not have any crystal structure for FER, and we still do not have the FER structure. But my talented graduate student, Matt, actually just wanted to computational modeling, even though I do not have much experience in that area, even though a long time ago I collaborated with some computer genius. He basically predict which residue we need to change, and then I basically made all the combination of the saturation mutagenesis mutant, and then screen 20,000 different mutant by hand. And that's very painful for me, and then that trauma actually prevent me from doing protein engineering. But this time, Matt basically used very nice approach using computational modeling and then predict the specific residue we need to change. And then actually when we change, let's say 20 different amino acid in one residue, he predicted, and another 20, another 20, so total probably not 100-ish or not 20,000, we could easily found and identify or discover new sensor that's specific to one compound, not the other, saving a lot of time and resources. So I'm very, happy. And he basically independently did that project himself. And that's why I'm so excited to working with young people like you, because our future is basically depending on you. So as I said, also based on our experimental data, we provided novel insight into the uncharacterized structure of COR, enabling further creation of sensor for additional neurochemicals. We actually currently collaborating with many different groups to develop wearable sensor. So think about future. One day your iPhone watch or whatever watch basically detect even neurochemicals. So when you wake up, your watch kind of tell you today your mood is good because your neurochemical level is very stable and so on. So that is kind of my dream. Or your watch also tell you you have some serious problem because your neurochemical balance not that great, also that's imbalanced. So do something that watch will tell us. So that is kind of what I want to do. And this work is basically first step for that goal. And we are currently developing many other neurochemicals, specific sensor, including dopamine, some other neurochemical. Okay, so next project. My idea is to develop so-called kill switches that allow engineered bacteria to self-destruct once they accomplish their mission. My idea is very simple. I'm a big fan of movies, and you may remember or not, if you are too young, Tom Cruise from Mission Impossible. Actually, he does not wear glasses, but what he actually doing is he got the glasses delivered to him and then just wear temporarily and then reads the secret message through mission containing glasses. And once he finished reading it, he throws them away and the glasses self-destruct. Same for my microbes. Once mission accomplished and then self-destruct, because this is the genetically engineered microbes and we don't know the consequence of this genetically engineered microbe in the real environment. I'm talking about our outside environment. So when you engineer probiotics, there are many issues to consider, including genetic stability, antibiotic resistance spread, loss of function mutations, and environmental release of the engineered microbes. Optimal biocontainment method would allow on-demand removal of probiotics from the gut and prevent any growth once they are released into the environment. Currently, there are some methods for biocontainment, for example, using auxotrophy or environmental signal inducible kill switches. However, auxotrophy could limit microbes' survival chance in the gut where the necessary nutrients or synthetic chemicals are missing. Also, temperature-induced kill switches have been demonstrated. I was also scooped again, but their performance has not been impressive so far. To address this issue, we have developed multi-input kill switch. First, we use an N-hydrotetracycline inducible sensor to develop kill switch. The reason why we chose ATC inducible system is that first, it is functional in diverse microbes, allowing for broad applications. Second, ATC is not present in natural diets, preventing false killing. Last, there are no known negative effects of ATC on animal health. We chose CRISPR-based killing mechanism because there are no non-homologous end-joining pathways in most microbes, allowing broad applications in many microbes, and CRISPR is also functional in many microbes, enabling generalization of our approach. We first checked the effect of guide RNA target site copy number on killing efficiency, or fraction viable, that is defined by induced colony forming unit divided by uninduced CFU. In other words, the lower fraction viable, the better. 0 means completely killing, induced killing. 1 means no killing at all. So we are kind of thinking about how this number become lower, and then NIH guideline is 10 to the minus 8. That means only 1 cell or fewer cells survive out of 10 to the 8th cell. However, as you can imagine, our killing efficiency was not so great, and we found most escapees are due to mutation on the Cas9 cassette. And after that, we literally fighting against mutation. Every single time we solve some problem, another mutation happening in other regions, including guide RNA, cassette, and so on. So as you can imagine, these bacteria do not want to commit suicide, but we keep saying or order, this is time to die. So this is constant kind of battle between us and the bacteria. So to make a long story short, as I said, the 6 years of failures and successes, we saw complete elimination of kill switch strain in mouse model. Here, red is our kill switch bacteria, and green and gray is our control. Control strain stay high population, and red one just completely gone. So again, my talented student Austin been through many failures and successes for 5 years, more than 5 years. And that this project actually initiated by some other people, and they actually graduate. And then Austin at that time knew this is a very challenging problem, but still ambitious enough to join in this project and then execute, even though made many failures. So I'm very grateful, and then I'm very proud of him. And this is the masterpiece of his work, and also masterpiece of my work as well. Unfortunately, we submit this one to Cell But Cell said, I mean, Cell generator said, this one is already demonstrated, so not sensational. So same thing, from the Nature Biotech. But we can actually, they kind of recommend some, additional journal. So we are kind of currently under review and actually final review. And this is a matter of time. I mean, this one is released, I mean, into the public, probably this month as an accepted paper. So finally, we also incorporate our temperature sensor to build 2-input kill switch where probiotic cells will self-destruct once they are released from the human or animal body. As I said, body temperature is 37 or plus minus 1, depending on your health condition. And then once bacteria out, the temperature typically probably is 34 or lower. And the bacteria kind of trigger the switching and basically kill themselves. We actually demonstrate complete killing again in vivo using mouse model. While I do not have the time to share the data or too lazy or busy to update my slide. So as I said, this paper under review and then hopefully this month out. So please stay tuned. So future direction include combining this kill switch with the application relevant functions. We can replace the temperature sensor, for example, with our other sensors I showed earlier, and then make a microbe self-destruct. So imagine, for example, I'm crazy, I'm thinking about kind of deploying my bacteria once all the safety feature confirmed, but still I can worry about, this bacteria could do something crazy in the environment. But to clean up our environment, we may need some bacteria deployed. And then let's say bacteria clean up our environment by eating all the toxic chemicals, including phenol or benzene, or even some plastic waste. And then once they clean up everything, unfortunately to them, I need to make them self-destruct. To do that, you could basically use some chemical sensor that detecting whatever waste, and then that bacteria will commit suicide, basically sacrificing them by cleaning up and then basically killing themselves. So that is basically one idea my lab currently pursuing, supported by EPA and USDA and NIH grant, as I kind of mentioned during my earlier presentation regarding probiotic. So I have 20 minutes each. So I kind of switch gear to another project. I call it metabolic engineering and systems biology. I envision our future as follows. We engineer microbes that takes waste as input and generate value-added products as output. One of the critical waste is plastic. And I believe this now is cliche to say plastic problem is critical. And our goal is not such beautiful PET recycling. This is not my goal, but PET upcycling. That is my goal. Our process is a hybrid process consisting of a hydrolysis system to generate TPA, one of the monomer, and EG, ethylene glycol, that's another monomer. And then microbial conversion of this into value-added product. We are interested in waste PT upcycling because waste PT represents 8% of global solid waste. I'm not talking about the basis is the total basis is the global plastic waste. I'm talking about global solid waste is the total, including metal and so on. That means 8% of global solid waste is huge amount. And then PET basically represent 8%. Because I mean, we are spoiled, we kind of using PET bottle, water bottle, Coca-Cola, whatever. And the current PET chemical recycling suffers from the relatively high cost of hydrolysis, monomer purification, and its repolymerization compared to the virgin PET price that's typically around $1 per kilogram. In contrast, our process uses a novel microbe, I call it magic microbe, that is flexible to potentially produce any desirable products, allowing us to cope with the market price fluctuation. Today, I will demonstrate two higher value products as a proof of concept. Probably I only show one slide about the carotenoid. There's a very high value compound up to several $1,000 per kilogram. There are two main challenges, of course. If there are no challenges, that's boring. And also probably I already solved the problem. I'm just a bit tired today and then just enjoy my life in Hawaii and then drinking cocktail in the beach. But there are many challenging problems we need to solve that make me excited. The first one is the efficient PET hydrolysis. State-of-the-art technology uses a purified LCC enzyme variant to achieve 90% of hydrolysis in 9 hours at 72 Celsius, and then make the PET again from the purified monomers. In contrast, we use NaOH hydrolysis without adding any additional catalyst to achieve similar yield at higher temperature for 0 hours, and then use our microbes to produce value-added chemicals without any purification step. We did some preliminary techno-economic analysis to obtain 90% monomer yield, show basically that data basically show both technology would similarly cost. For your information, the state-of-art technology making PET from PET actually currently undergoing in the French company, but I believe that is also a little bit not viable in terms of economics because that costs a lot in terms of purification and so on, and then making very low value compound. So that may solve the problem of the environmental pollution, but not necessarily creating value in the market. But however, my technology may solve both environmental problem and also sustainable chemical production, because my compound carotenoid currently produced or extracted from food, including carrot or tomato, by many Chinese company. But to me, that's kind of problem because extracting high value chemical from food, we should stop doing that because many people in developing country, including Africa, many people die every day because they don't have enough food. And one of my student, Tatenda, as I mentioned earlier, he did not have the electricity. not alone food. And then some other country actually extracting compound to make money out of the food. At the same time, many people die in the other side of the world. So my goal is currently solving those problems together, not using food to extracting those chemicals, but using waste, PT, and then making those chemicals to completely displace the current practice. The second challenge is the efficient monomer bioconversion. The main biological issue is that only a handful of reports demonstrated microbes capable of growing on both TPA and EG monomer. In addition, 2 more NaOH per 1 more released TPA is required to maintain pH 8. So the resultant media would have very high osmolarity, limiting high TPA and EG loading. The TPA is actually diacid. So as soon as TPA released from the polymer, that make the pH low. So you need to add NaOH to utilize. So that's what I'm talking about right now. Again, the state-of-the-art technology demonstrated PT to PHA biopolymer conversion using purified LCC, a little bit less efficient enzyme to degrade the PT into TPA and EG at 70 Celsius and 24 hours now, not rather than 9 hours. And then even lower conversion percentage, that's the 50%-ish. And then use the evolved Pseudomonas strain to produce PHA, the biopolymer. In contrast, our technology requires neither purification nor sterilization of the NaOH hydrolysis product. Importantly, our strain use a 1 order of magnitude higher monomer concentration despite high salt levels due to use NaOH and HCl for neutralization. Let me introduce our novel high salt tolerant Rhodococcus strain named RPET for the first time. What a cute name, right? We identified this in a bacteria by screening many wild type and mutant bacteria out of our mutant stock. And actually we have the 2,000 different bacteria and mutant stock in my -80 freezer. And we basically tested Pseudomonas and Rhodococcus, and they are mutant. And then this bacterium actually show up to 37% biomass yield per used PT, utilizing both monomers as sole carbon sources. We did not add any glucose. We used just hydrolysis product to make this bacterium grow. We have been developing many genetic tools for a related species called Rhodococcus opacus for many years. Unfortunately, these tools are also functional with some modification and optimization in our RPET, making engineering of this bacterium, the novel bacterium, possible. Interestingly, we observe significant beta-carotene production from the PT hydrolysis product when they are fed to wild-type RPET, especially under nitrogen-limiting condition. This result is consistent with other Rhodococcus species data. If we provide glucose and no nitrogen, they typically produce some carotenoid, but in our case, we only provide PT hydrolysis product, and then under lower nitrogen condition, and this bacterium naturally produce beta-carotene or lycopene if we engineer the pathway a little bit. And I believe this is the first carotenoid production from PT hydrolysis product. But I also don't want to sell, also don't want to provide hype. The current titer is very low, only up to 10 milligram per liter, or our best, fermentation situation. So with that, low titer, there is no commercial application we can consider. So my student and postdoc, including Jinjin, the postdoc, working very hard every day, even weekend, to improve this productivity, because we believe, and he believes, this is the really important problem to solve, including potentially climate crisis, and I will kind of mention a little bit later. So in summary of this topic, we discovered a novel salt-tolerant microbe that grew well up to 0.3 more TPA and EG. And in addition, we built its metabolic map for PT utilization by comparative genomic and knockout analysis, and demonstrated that high-value carotenoid production is possible from PT hydrolysis product. In parallel, we are also performing protein simulation assisted directive evolution to engineer mesophilic and thermophilic PT degrading enzymes. Again, my math student actually working on this project. Also, we are now team up with the computer genius in terms of protein engineering. And I believe this approach complements our NaOH hydrolysis approach. And actually, we observe PTAs containing lysate actually supported RPAS growth, and also identify mutation target for improved PTAs activity both at ambient and high temperatures. I wish I could share this data with more time, but I wait for the next opportunity or through publication. Actually, this one also under review in high impact journal right now. So hopefully stay tuned and we can cover this one further. So I believe I need to skip some of the thing. Okay, I already deleted that slide. So these are the summary of our current project. So we have the currently, actually, I show 6 projects, but we have the 7 active projects currently ongoing, including Probiotic project and kill switch project, and the plastic or waste upcycling or utilization project. And the last part, RNA engineering, I could not show, but that's basically metabolic engineering application of small regulatory RNA. And we currently working on this project. And actually, we working on that project for many years. So with that, I'd like to thank all my lab members, also my 2 advisors, Chris and Chris at MIT, for all the advice during my academic training, 10 years ago or 15 years ago, and also all the current funding agency, as I mentioned earlier. And one announcement to make, I believe some of you already know, but if you are interested in attending this event, please send me an email. For example, the SynBYSS seminar series did kick off on August 26th with Francis Arnold, of course, the Nobel Prize winner, my academic grandma, and also NSF, EBRC, Biomate directors. And on September 2nd, with Jeff Gordon, the microbiome father. Also more with all the editors, 11 national academic members, and many US government officers. It's weekly and more than 2 years via Zoom, and also this is the free for everyone. In-person meeting will be in Hawaii, which is not free, but it's in Hawaii. And then I will invite all the alums of the speaker, invited this, SynBYSS online seminar. I will invite them to Hawaii, and then they will have the in-person conference once COVID situation become better. I also became the YouTuber because many people asked me to share the video, and then people kindly allow me to share the recording. So this video already out, 12 video already out, and then now more than 10,000 views as of now. Also, I'm a council member of EBRC, the vision providing organization for the entire world and the White House and funders. And we are organizing climate sustainability road mapping workshops that are open to non-EBRC members. And the next one will be December 13th, free, everyone invited. More detail here on my seminar series, also, all the website, EBRC website, and so on. Also, I regularly kind of tweeting about this seminar series, so just check it out. Something to share. Actually, I slept with this huge guy when I traveling a 10-day trip for the seminar, 8 different seminar a few weeks ago. And this guy, oh my God, spider, the biggest one I ever seen in my hotel room. Actually, when I saw it, of course, however, I saw the web first, and then the spider. Of course, no dollar he pay or she pay, so no sleep inside my hotel room. This is actually outside my window. Then I realized how much we are connected. This guy make us connected. We are all connected. Without connection, many do not function, including the web, guitars, trains, and the most importantly, human, or all living world. I enjoy my walking from Boston University to Harvard University when I doing seminar a few weeks ago with the unbelievable warm Boston November. And I would say climate change is no longer change, but crisis or emergency. I hope the COP26 and our technology, or you, bring the real solution to climate emergency. And God bless all of us. So another thing to share, I'm the luckiest guy to have the Crystalla Preder as my lifetime mentor, but I have not been as a great mentor as she is. For example, one of my students, Tatenda Chopra, was a victim of the discrimination, but I had been a coward who could not speak up against the leadership people who harassed me to cover it up right before my tenure evaluation. I was coward and saved my ass for my family. I'm no longer coward. Luckily, I was able to at least prevent them from kicking him out of the university and US, and his dream came true by developing the Pfizer COVID vaccine for all of us. I'm super proud of him and myself. The last thing to share, I learned this by my experience, but it took me almost 50 years to realize this by heart. Jeff's quote touched me so much. Unfortunately, I saw this slide late July 2021. I wish I saw it earlier. I believe from the Dustin, a musician and brilliant storyteller at America's Got Talent, he said, quote, "Giving is the foundation of the change," unquote. I want to say we are all busy, and we have our own problem, our family members dying or pass away. I had my own personal medical issue for many years, discrimination, also harassment, but we can change this world together by giving our time, resources, attention, support, compassion, kind word, empathy, even forgiveness, all the S words. Of course, our love. Also, I would say the future is made by young people like you. I'm not a big guy, and big names like Jeff Gordon and Francis Arnold can give the vision to us. However, the real implementation should be done by young people like you, the future leaders. So dream high, and nothing is impossible. Our future depends on you. One last thing, I declare some conflict of interest. Thank you so much. I'm sorry. Now I realize I kind of passed my time. I'm happy to answer any question by email or some other thing. Just shoot me email. I'm happy to answer any question.
Thanks so much for such a unique, inspiring talk. We're honored to have you. And would you take questions on your SynBYSS Slack channel, perhaps?
Yeah, so in the email should be better because I'm not okay. the social media group. So email is much better. So I also need to respect my colleague. I already see over here, and hey, long time no see. So I also need to just shut up to give more time to him. But I'm welcome to any email. I normally check my email in the morning. So I will answer any question you may have. Any question, career advice, life advice. I'm getting almost 50, so I'm happy to share my experience. Even I could do chatting with those Zoom.
Mm-hmm. Yeah, thank you so much.
I'll stay, for a while, so I will stay on probably entire, conference, today.